Good afternoon, uh, my participants on this training course. You are welcome. I am Basu. I worked for Central Pollution Control Board since 1976 and retired on 2012 and joined CAC for last 9 years. I am working as an advisor to CAC. During my tenure in Central Pollution Control Board, I generally worked on various subjects, laboratory, then comprehensive industry documents to setting standard emission effluent, and also hazard waste management sites. Then at the fag end, I also being in charge of the National Water Quality Monitoring and National Air Quality Monitoring Program and Noise Quality Monitoring Program. So now today I am giving you the baseline data collection, which is the backbone of environmental impact assessment. Next. Generally, any scientific rationale, if you want to base a science-based decision-making process, you have to collect data. So the real world, it is basically a deaf and dumb. You have to ask questions through your experiment or give a deep observations which generate the data. This data has to be, sometimes people also go for model. That is this conceptual, that how this will be happened. And this model has to be reasoning and calculation and give is the prediction. If the prediction and the data agree and disagree, if it is agreed, then model is fit, and it does not, then model is true, false. So you have to have a data collection systems to see that what you are predicting. In environmental impact assessment, what is happening, you do not know what will be happened after the industry started its construction phase, operation phases. So you have to have a rely on, you are developing a model on water quality model or DO-BOD coupling reaction, which is a famous model, Treater-Phelps models. Also, you have a dispersion models in air pollution. And then you try to reasoning that it will be happen like this, you predict it. But if your data you collected, and this data collection and the prediction, if it is agreed, then it is a good, that hypothesis is accepted true. If it is not, then hypothesis is false. So past data and the data actually happening in the process of construction phase and in the process of operation phase, you have to see this thing. Not only industry, it is also in case of any big project, that is the urban system they are developing, very big housing projects, or also big project like dam construction, etc., or the railway track. These are all the things. It has to be, the basis is this data collection. Next. Why it is important, that I have already told you, to provide a description of the status and trends of environmental factors against which predicted changes can be compared and evaluated. Detect annual change, actual change to the environment after the project has been initiated. These are the two things, is the most important things. Next. Database, what are the database required? Impact on air, environment. It is the database required, air quality, and air pollutant is movement of the air pollutions, It depends on the meteorology. That's the wind speed in horizontally and mixing height. It is the vertical distance. You also in impact on air also encompasses noise. Then the impact on water body. In surface water body, what is happening? What is the nature of the water use in this riverine systems or lake systems? What are they use? Maybe drinking purpose, maybe agriculture purpose, maybe purpose of the bathing. It is a very important in Indian context where no one know where the organized bathing like Kumbh Mela, Ganga Sagar Mela. These are the things are organized bathing where lakhs of people are taking bath. Then the question is the hydrology. Hydrology is a very important part because hydrology will tell you the what is the quantity of water available throughout the year. In critical time, that is the summer time, what is the critical flow of the river? That is the very important in hydrology study. Because if there is no flow of the river, then pollutant, how pollutant can be dispersed? That is also not possible. And also, of course, dilution is not the solution of the pollution, but the cases of this, some kind of a aquatic environment should be there, which can break down the biodegradable product and others, or utilize the nutrients. Then the actual water quality, that has to be measures. In case of the groundwater flow, hydrogeological studies of the groundwater and the character of the groundwater, and also the groundwater use. That is mostly in India, the groundwater used for water drinking as well as irrigation purpose. Soils and lithosphere, nature of soils, that is soil quality and its potential, and agriculture use. That soil, that is the most thing. The fertile soil is there and it has a potential for agriculture, and agriculture is doing multiple cropping is there. That is the land is very important for the our food generation. So these are the things land has to be understood. Then ecology, the terrestrial ecology, the ecology on the land, what kind of thing, forestry, movement, these are things. And then forestry also take care of the wildlife propagations, aquatic environment, aquatic ecosystems, which is very important. More the diversity is there in aquatic ecosystem or terrestrial ecosystem, better is the stability of the ecosystem. Then socioeconomic study also has to be changed. And population and migration patterns. That if you are going for a mining, there will be migratory laborers, which has a clash with the existing population, a population who have lost the land for the migrations. What is their socioeconomic characteristics? And what are the sources of livelihood? Before the starting project, they may be dependent on the forestry or agriculture, and now they have been thrown away from the land. The owner is getting the things. Now, in a new act of the Land Acquisition Act, where the agricultural labor actually, who is not the owner, so this labor also, if this registered, and they are also gaining some benefit. So these are the things has to be taking care of the socio-economic aspects. Next. Now, baseline data generation, before that you have to see that what are the availability of data. India Meteorological Department, they have a century-old meteorological data is there. And that is available, they sell it. You can take that data also. If it is see that their data, the meteorological data of that place where they are located, their collection, that is within the 40 to 50 kilometers, it is okay, this data is beyond that, then you have to collect the meteorological data. CPCB, SPCB has a very big monitoring network. They have operating 700 stations on the National Air Quality Monitoring Program. Also, they have a COCAM, that is the continuous air quality monitoring stations are there. So these data are also available. And this data, if you process, you will find what is the gap, what are the data more required. So you have to go for a design of a monitoring program, then the data coming, data analysis, and then you have to go for a prediction. Same is the case of water quality. Water quality, public health engineering department and other department, they are giving you, the agriculture department, fishery, they are using, giving you the water use. That this is the stretch of the river. This purpose, a PhD will give drinking water, public health engineering department. Agriculture department will give you the what are the irrigation they are telling, and fishery. Some of the places irrigation is out of the divided into agriculture and irrigation. Irrigation department is separate. You can go for separate. Then groundwater quality, quantity and quality. Groundwater board have a very big network. Very old database is there. This data also available. Then Central Water Commission and CPCB, SPCB also doing for some quantity analysis, quality and quantity analysis. Quality, they have a nearly 5,000 stations, CPCB, SPCB network. Then the surface water quantity data, you will go for the Central Water Commission. They have a database. You process, you will find what is the gap, and accordingly you you program, a monitoring program you initiated, data analysis, and then go for the prediction. So you have a very good soil quality data. The state agriculture department has a good database. NBSS, National Bureau of Soil Survey, Nagpur, they have also district-wise soil quality data is available. This data you can process and find out that agriculture potentiality, this potentiality, and then there is a no, it is not a good agricultural land, it's a wasteland. You can go for a industry very jolly well, you can go. Socioeconomic population data is always you go for the sensor. And sensor data, you have to little bit correction is necessary because 2011, I think the next is that 2021 or so this data you have to yearly you have to connect it, and 2.5% or something like that is you have to every year it is increasing. So that collection is necessary. And the second thing is that you have to go for a questionnaire survey. This survey can be done reconnaissance because you send the question, villager will not answer. You go to door collection, or if you can go for a kind of a medical camp or some camp is given and they are coming because it is also helping them to have a medical checkup. At the same time, you can collect the relevant data also. Next. Now my question that how to design a water quality monitoring network. You want to do a water quality monitoring network, you have to design a water quality monitoring network. What are the design? What are the criteria? How you will develop the water quality monitoring network? Next. Water quality, what do you mean? Water quality means the physiochemical characteristic of water that harbors diverse aquatic flora and fauna. That means it has a aquatic flora and fauna and various uses for human being. So that means that not only the water quality in terms of nitrate, sulfate, but we also see that what is the diversity aquatic flora and diversity index you have to see. What are the phytoplankton? What are the zooplankton which are required? Some of the bacteria also, they are break down the BOD also in the river. And various uses of human beings. Already I have told drinking water purpose, bathing purpose, then drinking water with treatment That is also another category. Fourth category has been given the physiculture, etc. And five is the agriculture also. So these are the generally, and wastewater discharge, navigation also is coming to the last category. CPCB has done a categorization of these things, schemation, zoning, and classification of water body. There they have given A, B, C, D, E, and this classification is basis. On the basis, they develop water quality criteria. And on the water quality criteria, you know what is violated or not violated. Next. Now, this is the things has to be understood very carefully. Why ecosystem is river? Why ecosystem is important? Because if it is surface runoff is there, you will find some dissolved in inorganic nutrients. Carbon, nitrogen, and phosphate is building block of the life, right? That gave birth to periphyton or phytoplankton. Then phytoplankton is going to aquatic invertebrates. Then you can go to the omnivorous species, and then it is going to humans. This is the movement. Now you see this, there is a detrivorous species also. They are eating, right? And Piscivorous species, that is also eating. Then there is a detritus, that is the detritus has been taken by some heterogenic bacteria. They are also cleaning the things. And terrestrial plants is also there. And then this is the way the entire ecosystem, a complex systems, and a complete food web has been developed. And these food webs is the basis of the water quality. If this feed waste is there, if you are discharge wastewater, the heterogenic bacteria starts first working. The nitrogen, they are break down the BOD and making into carbon dioxide. And the phytoplankton and the zooplankton, they are taking the aquatic macrophytes, they are taking the nutrients and carbon also. So these are the things. And they are cleaning actually. It is the and if you are determined the very low load, if you can treat the load and give it to the aquatic ecosystem, they will change it. No river can be done with the treated wastewater. So you have a minimum flow, ecological flow, and this flow will maintain the ecosystem. That is why while drafting the Water Act, they are telling that to maintain and restore the wholesomeness of water. And judiciary interpret very nicely that it is the aquatic ecosystem has to be maintained. That is the most important thing, to have a flora and fauna and ensure the different uses of the human being. Next. Monitoring program for a systematic observation in order to draw inferences about the experiment or the phenomena for which it is designed. It is a systematic observation. That means they have stations from where you are collecting the water or on any things in air or noise like that. And then you have an observation that what is the flora and fauna is working there. And then you experiments. That is, you collect the water and laboratory experiments. What are the nitrates? What are the phosphate? What is the BOD? What is the COD? What is the solids? And then you can predict if this is the load, the dissolved oxygen will be like. The minimum dissolved oxygen 4 milligram per liter is required to sustain the aquatic ecosystem. Next. Systematic observation, a periodic observation with regular intervals. How often, that when to observe, frequency of observations. That means how often you to observe, once in a month, once in a week. That has to be, see, if the variation of the river is very high, that means spread is data, then your frequency will be increases. If it is not, once in a month. Our experiences say once in a month, it is very good because seasonal variation is taking place in water quality until abrupt, abrupt uh, wastewater discharge is there. Next. Observation in science means measurement. What to measure? Parameters to be defined. That I will explain. What are the water quality parameters to be defined? Next. The third component monitoring is location, where the sample should be there, where to sample, where the sample collection should be there. Next. So monitoring therefore location of sample, where to sample, frequency of sampling, how often, and what to the sample. That means what are the parameters. So where to sample, when, how often, what are the parameters. This is the thing is the monitoring. Monitoring. If you want to monitoring network design, this is the three factors. The three criteria has to be maintained. Next. So management action is that what information should be the monitoring program generates. That is what to be measured, what to be sampled, where, how often. That has to be seen. If there is there, if the river has a, if the wastewater discharge will be highly biodegradable, BOD high, then your sampling system will be different, BOD, COD. If it is a COD, BOD high, COD also high, and there is a TDS is there, then your parameters will be selected like this. So first of all, you have to go for a preliminary subtests. Obtain background information, reconnaissance. Reconnaissance is a very important if you want to do a monitoring program. You have to take a vehicles and go along the river, longitudinal distance. In case of Ganga, I have gone from Rishikesh to Diamond Harbour to select the stations, talking with the persons, different type of persons, PhD persons, and then one after one station has been selected. Then you adequacy of the monitoring data. Too much stations always give a good results. Lot of stations. At the camp, it is costly also. So you have to optimize the number of locations. You have to and make a map where this thing and GPS system. You select the station along with the GPS system. Then resource estimation. What is the lab requirement? What is the transport? What is the staffing and monitoring thing? Field may, what we will do in the field work and sampling, field testings method, hydrological measurements. Your hydrological measurement you have to do. For a small stream, a consultant can do, or in university can do, or a pollution control book do. Any big stream like Ganga and etc. You have to have a monitoring. The hydrological measurement will be on the flow meter. That is a must. Then laboratory work, physical, chemical analysis, lab test and procedure. If their lab should be accredited by NABL, National Air Accreditation Board of Laboratories. That that is must be certified. EPA under EPA Act, Ministry of Environment is certified. In that case, the certification, you will find that in EPA certifications or state board certification for water quality monitoring under Water Act they can do. You have to have a NABL accreditation. Otherwise, they will not give. Because I was there, I know that it is the procedure. First, you enable, and enable number of parameters has to be set. In enable system, you have to go for analytical quality assurance. This program is done by Central Board for the State Pollution Control Board, also EPA recognized laboratory. Then data management, quality control, storage, statistical analysis, and reporting. This is the entire management actions. Next. So you have a more or less 8-step setting water quality monitoring objects. assessment of resources, laboratory facility, transport, manpower, reconnaissance survey, that I am telling you, map of the area, background information, human activities, whether that huge stretch of the land is only agriculture. Maybe some places where industrialization is there, some places urbanization is there. Then you have to find out potential pollution sources, where sewage is coming, where industrial wastewater coming, where agriculture runoff is coming, hydrological information, and water related thinking actually. Next, network design, selection of sampling location, optimum number of location, parameters, component frequency and component to be some water, sediment, biota, sampling, representative sampling. This is very important. What is representative? I will come. Field testing, some of the parameters you have to do at the field also. Sample preservation and transport. All I will discuss it very clearly. Next. Laboratory work, laboratory procedures. That procedures has to be laid down either APHA, American Public Health Association, or BIS, Bureau of Indian Standard. They have their, that is their, under the purview of them, they are doing. Central Board has given published a book that given the outlines and guidelines for water quality monitoring. Then a quality assurances program, internal accuracy and external accuracy. That laboratory should be a very good laboratory. That I have to say. And data management, storage, statistical analysis, presentation, interpretation, and reporting. Storage is very important. This storage, if you keep it with you, any organization for a, suppose you have studied a water quality of a streams or of a river, then you must store the data for a long time. And that storage you have to be maintained actually. But this will help you. Also 5 years after again you have to do for that places, then this database will be helping you. That is your past data, you will know that what is the changes happened. Next. So what is the objectives of water quality management? First of all, the baseline condition in the core systems. What is the baseline condition? That is the must while giving doing your EIA report. And find out the place where the deterioration takes place with respect to the standard, aquatic standard, which is given by Central Board. Identification of any water bodies in the water system that do not meet the desired water quality standard. And that I have told. Contaminated areas, these are the hotspots. And determination of the extent or effects of specific waste discharge. If you are discharging this wastewater, what will be the impact of that things? That you have to predict, right? That is Titterfell's equation is there. You can estimate the K1 value and K2 values. That means your deterioration, the K1 is this, and K2 is the reaeration. That is what is the reaeration. Reaeration values also you can calculate very easily in the riverine system. And K1, there are lot of studies have been done. So what is the BOD breakdown? These things is there. And you can get the specific and go for a Titterfell's bond. Simple Titterfell's bond. And estimation of pollution load carried out by a water course system or subsystems. Evaluation of effectiveness of the water quality management intervention. That is there. What is the intervention is required for water quality management? If you are discharging wastewater, what will the intervention is there? Then how much treatment is required? That has to be seen. Water quality guidelines and standards for specific water uses. That is there. If it is if your downstream has a sensitive water use, a drinking water source, it is a very important for you. Then you may discharge downstream of the water. That was a very good thing. Then you can save that here is the intake point of the water for the urban systems. or rural systems, but and you are below these things, then discharge, if there is one or other ways, if the discharge disturbs, it doesn't disturb the water quality of the desired water quality for drinking water source. Then development of water regulation covering the quantity and quality, development of water pollution control program. That is the case of the pollution control board. Next, these are total objectives I have given you. There are 4 types of station is there. One is the baseline. At the upstream, you can do it. But in the case of Ganga, in your states of Ganga, you will find that there is a upstream station is right above the Rishikesh actually. Flux stations. This is the tributary irrigation drains, stormwater drains, or this stormwater drains carries the water. That means it is flux that the quantity load is discharging to that main river. This station is called a flux station. So your discharge point will be a flux point. Suppose you are discharging, you have to maintain that what is the flux you are discharging. A monitoring train means to show how a particular point on water course varies over time. This is a trained analysis. Impact means just downstream of the mixing zone of the wastewater. The wastewater discharge, it is mixed to the things, and that is the impact station, which is very important. If it is good, a good consultant may suggest the project proponent that this impact station should be automatic station, real-time station, so that continuous data they are observing that they are not discharging. If they are discharging, so if somebody goes to the code, they can say, this is the view. See, sir, we have done. So I have not, my discharge doesn't create any problems. Next. It is there, baseline, like the flux tributary is coming, or drain is coming, or your discharge point is coming. And just downstream of the flux, the impact station is there, global is there. And then you will find some stations like these here. It is here, this train station is there. There also a train station where a little bit here. And global flux is not required. It is required only if you are discharging, your industries here, and the entire wastewater is going to coastal ecosystem. That time you should do it. Next. Now, what is the parameters? General health, pH, DO, conductivity, temperature. At a general health, if you know the DO, that means the oxygen level, you know. If it is a 4 milligram per liter, it is okay. Conductivity means you're giving what is the salt is coming in the river. pH, that means acidity and alkalinity, and a temperature. Ions, when you are going, then you are going for calcium carbonate, magnesium as carbonate, calcium carbonate, sodium, sulfate, bicarbonate, chloride. That means hardness and ionic behavior, salinity, is given by the ions. There is one ion is fluoride, right? This fluoride is a very important case is there. This fluoride is very important because fluorosis is coming. If it is a certain 1 milligram per liter above, it's there, then the people should be concerned that there is there. Some fluoride is required for the teeth, and some excess fluoride is decaying the things. It is more important in groundwater cases, but in surface water also, fluoride, arsenic also coming as a threat to the water quality. Next. Then free ammonia. That is the free ammonia. If free ammonia is there, fish will not survive. It is giving the fish toxicity. Then BOD, COD, that is, and fake alcohol, BOD and COD is organic matters. BOD is a subset of COD. BOD means biodegradable wastewater. COD means biodegradable wastewater plus persistent organic. If COD is more than BOD, COD always more than BOD, but if COD is very big compared to BOD, then the persistent organic is there. It may not alter the things, but it will create destroy the ecosystems. So that this is pesticides industry, bulk drug industry, they have to be careful about the COD BOD ratio. Then fecal coliform. This is giving a total coliform. These are two indicating parameters for pathogens. Generally, in Indian river, pathogens are there everywhere because we have a huge population. Open defecation was there, although there is a Swachh Bharat program. It's a good program. I'm thinking this is one of the good program the government have initiated so that the fecal contamination will be less. And fecal and total coliform everywhere in India. But the fortunate part is that it cannot survive for a long time because of the sunshines. Then nitrate and phosphate, these are the two parameters for the eutrophication. And heavy metal toxicity and pesticides. Heavy metals is coming to some extent where industrial toxicity, either say the electroplating is there, the hexavalent chromium, etc. is a toxic chemicals, chromium, total chromium, these are coming. And then pesticides is generally coming where the agricultural runoff is the excess pesticides here, or pesticides companies there whose residual pesticides are coming. That has to be maintained the standard pesticides industries. Next. Now we have to go for a baseline data collection. That is the frequency. 4 times in a year, if it is a perennial river. All the things you don't have to go because basically it doesn't change. General parameter is a color, odor, temperature, pH, and fecal coliform, electrical conductance, dissolved oxygen, total dissolved solids, and then ammoniacal nitrogen, nitrate, and nitrite, and nitrogen oxide. This total kernel nitrogen, if you take, and nitrate you take, it will sufficient and free ammonia. Then demand means oxygen demand, BOD and COD. Also you have to measure ions and other inorganic, fluoride. If fluoride is there, you have to say once or 2 times fluoride is there, not there, then you don't continue for a long time. Now, in case of the, this is the general parameters 4 times in a year. And of the year, rest of the year after the pre-monsoon sampling and etc., you give a small parameters. Ozone, temperature, pH, ECDO, and this is there. 4 times a year you do all the things, and the rest of the time you go for this type of things. It is a perennial river. Next. Trend analysis, more or less same parameters you have to choose. Pre-monsoon analyze, parameter 25 listed for baseline monitoring. That you have to do. Ammonia, organic nitrogen, measure ions, microbiology, then this. Pesticides, you will see what kind of pesticides used in the agriculture, that area, and accordingly you select the stations. Accordingly you select the parameters. Not all the pesticides you have to do. Toxic metal also you have an inventory. These are all things you can talk with the state pollution control and persons, the regional offices. They will give you the actually good database. If you talk with them and they you interact with them, and you will get a good database that what are the toxic metal you are indicating. Next. Now, how to select a monitoring station? As I have told you, you have to go for a reconnaissance survey. Then you have to go for a mixing zone where the flux stations discharge and the main river is totally well mixing. Depth. If it is a very deep river, then you have to be one or two station in vertically, one or two water sample collected on that station vertically. But generally in Indian river 1 meter depth is there. The minimum distance, that is the things you have to see, how much distance have to be kept. Next. Location map you have to see when you are going, background information of water body. That means what are the where the major settlement is there, where the district town is there, where the metropolitan town is there. These are the things. Artificial potential polluting sources. What are the polluting potential sources? If there is a cities are there or towns are there, towns wastewater discharge you also find out. These are very important, these things. Actually, you will say that I am not the alone is discharging wastewater. There are so many parties are there. And if I go there, what will be the aggregate situation? That has to be understood very clearly. Water abstraction, quality and quantity. If the water abstraction is there, huge water is abstracted for drinking water purpose, for irrigation purpose, then what is the downstream's impact is there? What are the dilution available in the river? And if you are a big industry, suppose thermal power station, you are also abstracting, you will abstract a huge amount of water. So that then what will be the fate of the downstream? What will the flow of that thing? And how much you will be recycling? How will best use of the water? These are all things has to be taken care of when preparing the EIA report. Use pattern I have told, water flow regulation is there. There are, if dam is there, they have a water flow regulation is there. You will also talk with the dam people. Then when you are discharging, storage, how much these things, they are discharging also. Next. I have given these studies actually in the Kali River. When I studied, I have seen that, you will see that all the things is this color, it is a wastewater discharge actually, right? And this here, the Kali rivers, the Ganga water is coming. And these are the stations, impact stations, mostly impact station, because downstream of this, it is a Mirat city, it's a kind of another city, Hapur and etc. So these are the station I have given, right? So this is the things. So you have to select, you go the reconnaissance, find out each and everything I have calculated. You have seen this thing. The nalas, all the drains I have calculated. How much quantity of wastewater is going and what is the BOD load or COD load or TDS load. Accordingly, we have done this things, right? Next. Mixing zone is very important. Very important. What has happened? Once one fellow, here is the L1, that is the reference point, and it is the impact station is the L2, right? And L3 is the flux. So Q1 C1 plus Q2 C2 equal to QC. Here is the QC, right? Now one fellow has to collecting sample here and reported 30 milligram per liter BOD. In river, it is impossible. And it was came to the parliament question was there. Then I have gone there personally because the concerned chairman and joint secretary told you go, what is happening. Then he told there was no boatman, so he collected here. Actually, you look, here is the river is flowing. If you take sample from here, you are actually collecting the sewage or industrial waste. Because here the things are mixing totally. River water and there. And here you have to take the sample. Here you take the flux samples. Here are the things. And if you take the sample, river sample, it is not a river. It is the river wastewater is coming and mixing. This is the mixing zone. Now you tell me, you will tell I have no wastewater discharge when I am doing the EIA report. What is rhodamine B you put? It is a red color. And it will come. It will come and then you will find that somewhere it is mixing. This I have done in the case of some of the rivers in Andhra Pradesh, one river, I think Gautami. I have done these experiments with the help of Mary. This is a rhodamine B experiments is there. Rhodamine B is discharged, is a red color, and after 5 minutes, it's mixing like an impact station, and then we select the station here. After few minutes, after half an hour, the rhodamine B, this color has been faded. Nothing is there. Next. So this is a one guidelines has been given by WHO that average width is 5 meter and mean depth is 1, 2, 3. So what is the estimated distance? You see that thing. The estimated distance, if the depth is there, 0.02 to 0. Quickly it is there, right? If depth is not there, it is flowing actually. So this is the thing. In the case of 10 average, if the mean depth is like this meter, then you will find that things. If the low depth, your mixing time is stacking and it is only 0.7 to 0.5. You have to calculate and find out the mixing zone and depth. Next. Now, excuse me there. Average discharge is less than 5. It is a small meter cube per second. Then it's a small stream, number of sampling points will be 2 and number of sampling depths will be 1. Only 1 meter. And 5 to 140. Then you can go for a 2 sampling depths. But generally, I am telling in Indian river is such that it is only 1 meter is sufficient for you. That I have calculated because most of the time the depth is very low. A large river is there, number of sampling 4, but is also I have found a 1 meter is sufficient. You take a 1 meter and go. Large river is a 1000 meter cube, you need more than 6 stations. This is dependent on reconnaissance survey. Next. Next. Now I am giving the sampling program. Now population, suppose you are taking a huge flow of water is going. Once in a month you are going and taking one sample and predict the population. This sample must be representative. That means your collection in such a way that every element of the collection has the same chance of being selected. That is the most important thing. That means in the middle of the river, mixing zone is there. Where the mixing zone is completed, you take the samples. Next. There are 3 types of sample: random sample, composite sample, and graph sample. In case of this, if you are doing, it is a graph sample. That is, you just every month you are taking and taking once time. But if you are going just the impact station which you have predicted from your wastewater discharge, that station, you should go for a composite sample. That means every time you have a different DO. So you know the diurnal behavior of the DO. If the nitrate is high, suppose nitrate is high, phosphate is high, then the fight, and if you see that algal boom is there, then what is happening? At 12 o'clock in the noon time, you will find a huge DO, even about 12 milligram per liter. And that evening and early morning, the DO is almost nil. So this is called the diurnal behavior. So in the lower things, you will find the composite samples. Next. This is their composite. So say 8 o'clock, you are taking the sample, mixing here. 10 o'clock, you are sampling the sample, you mixing here. And then 12 o'clock, you like this. So 2 hours interval, you are taking some samples according to the flow, and you are giving the composite. and grab samples. Just you take one sample once in a while, and that is sufficient. And it should be always air, right? It is the middle, not from the hair. Next, in situ measurement. Which of them you have to measure in situ? That is on the field. You cannot do it outside. Color, odor, temperature, pH, conductivity, and DO fixation. That means you take the DO bottle and fix it with these your fixations, and then you keep it, and then come in the things. All you do by DO meter. Next, color may be light type things. Light brown color. Brown color is that some kind of a contamination is there. Dark brown, light brown color. Light green, green. Light green is the some amount of a very thin amount of phytoplankton or these things are there, algal things are there. With dark green, it is a eutrophication is taking place. That means lot of things are, which is bad. There is a clear and other specificity. I have seen a very crystal clear river in the upstreams of Himalayas, even in Mandovi River in Goa upstream. Oh, wonderful, absolutely clear. You can see the bottom also. But generally, you will find in Indian river as a light green color, right? And brown color also we have seen when their distillery wastewater is discharged. Next, odor free. You take the sample and then shake it and then put it in the nose. If you are not smoker, I have one girl. She was nothing. She has nothing, so she was not taking anything. So you see, smoke and tell whether the egg is soapy, fishy, aromatic, whether something is there, unpleasant, color-free, alcoholic. So everything you can find out. You see, it has been testing like this. First, you have setting your nose by different color, different smell, very light diluted in the laboratory and fix it like this, and then come to the river and take immediately shake and this smell, and it can be done. Next. So what are the things you should take? I am telling itinerary for the trip, where you will, where your station, everything that is there, and then area map, ice box filled with ice or step, BOD bottles, special sample containers, bacteriological for heavy metals. Heavy metal you have to dip some acid also. What are the preservator you have to dip, tissue paper, sample identification forms, right? You have to do these things. So a spirit lamp is very necessary for total coliform and fecal coliform. You have to have your own drinking water. Some places in river you have no habitat. So you have, if you are drinking water, you should take with this thing. First aid box, and then sampling, weighing bottle sampler, weighted bottle sampler you have to see. There is a lot of sampler is available now. Rope, sample container, thermometer, pen, pencil, marker is very good for, because we'll write down in the bottle where the things are there. Matchbox, because of the spirit lamp you have to lighten. Knife and gloves, and if it is a, if you are doing the flux station is a sewage, you should not do by hand. You should do it with the gloves. Pump and accessories is not required, but sometimes we take whenever the riverine system. Generally, we take the samples from the bridge. Railway bridge is the best one. It has been well measured bridge actually. And railway bridge may you will find the length is there. That is flow meter. If you have a flow meter, you can measure it because length is given in railway bridge. You have seen the railway bridge. There is a measurement system set there. And good national highway bridge also the scale is there. Height of the things are there. How much water height from the bottom. That was scaled there. So if you flow measurement is there, so meter, you know the length of the things, you know the meter per second, the flow meter, and meter into meter square, and then height meter cube per second. That way you have to calculate. Next, rinse the sample container 3 times with the sample before it is done. So you take, don't rinse with other water. You take it, the bottle, leave a small air space in the bottle to allow mixing of sample at the time of analysis. Properly leveling is required. Leveling is the most important. Complete the sample identification from each sample, and sample identification form should be kept in a master file at the laboratory where the sample is analysis. Next. Sample will be collected with a weighted bottle or DO 30 centimeter below the water surface. Samples from reservoir, it will be always outside. That means it is going discharge the canal, you take the canal. And if there is a regulated, then you go for a regulatory structure, upstream of the regulatory structure, input there. DO is determined in a sample collection and DO bottle. You have to a DO bottle using DO sampler, and DO bottle will fix in DO sampler and put it there, and then take the samples. Immediately after collection, you use the reagents so that it is fixed. Or you can use the DO meter. Now a very good DO meter is available. Next. Sampling code number. You have to give a code system. Central Board has a very good code system. You can follow that code system also. Date and time of sampling. Source and type of sample. Pre-treatment or preservation carried out at the sample. Any special notes for the analyst. ये बारिश हुआ कि नहीं हुआ, last day में बारिश हुआ, these are all the things you have to be noted. Next. So container is that on-site analysis pH bowl or container, yeah, plus minus 200 volume is there. For SAS, TDS, is 1000 milliliter volume on glass or polychloride दे दीजिए. Then in USN COD ammonia nitrate, then you have to go for glass and P 500. Phosphate is P. Polyethylene bottle actually. BOD is 1000. Coliform is given. It is better to take in the glass, and the sterilized glass. It is better. Heavy metal, you can take the glass or you can polyethylene, but you have to have a preservator, that is nitric acid pH2. Mercury, you will go and you use nitric acid pH2. Glass, pesticides, glass or Teflon, it is these things there. Standard things. I have given all the things to you so that you can, when you are going in the field, you will get these things and have some fair idea how to do all the things on the field. Field is very important. Sampling is the most important. 80% credit goes to the sampling. Next. This is a chart, you know. This is the chart you can fix. And this is the standard chart. All over the world, GEMS, Global Environmental Monitoring Stations. Throughout the world, the global GEMS stations is operating. And this is a standard thing. It is happened in CPCB's SPCB monitoring network, is also CWC is following. This is the best systems. So data, code, then thing there, then what are the parameters you are collecting in situ. All the things has been written here. Loader, color, everything has been written. It's a very good data sheet. Next. You don't have to invent a new data set. Then measurement of the methods that I have given the significant of figures after decimal. Too much big. 4.5434. Your weighing balance has to be things that DO meter It is a one digit, 6.1, 6.4. This is like the DO you have to give. Conductivity, 320. 320.4 is nothing, meaningless. 320. You write down this. If 320.6, it is 321. Dissolved solid also like that. Where the bigger things are there is there. Phosphate and orthophosphate is coming very less. That's why four significant figures, 0.4341, right? Then all the things I have given you, that what are the method, measuring method has been given by the Bureau of Indian Standard or APHA, both of them written like this. You have to follow this method. No other methods will be acceptable before the law, right? Because code will ask what method you will follow. Next. Now, data valuation. Checking entry of the data. Suppose you analyze, you take the sample, you analyze. Now, entry of the data is wrong because it is typing by a typist. He has nothing to do here actually. If you are doing by yourself, it is okay. But always data entry you have to change. Detection limit का ऊपर है कि ना? अगर detection limit has been given, every method has a detection limit. And below the detection limit, you should not report. You say below detection, BDL. Then expected range, you know this type of range, a very good water quality monitor for a long time who is monitoring, he will know that what is the expectation. Okay? Too many significant digit is not required. Checking if data is physically or scientifically possible, general check. You will know dissolved oxygen 12 milligram per liter is not possible. It is not at all possible. BOD is more than 30 milligram per liter in a river. It is not possible. The total dissolved solid 3000 milligram per liter. It is not there. It is be around 500 to 800. And in India, 500. In a very good river, it will be 100, 200. Right? In Scotland, it is a 50 milligram per liter. The checking correlation BOD COD relation, EC and TDS, there must be a correlation. There must be a correlation. And cation and anions must be a balance. Ionic balance should be there. Total iron must be greater than the dissolved iron. Total phosphate must be greater than the dissolved orthophosphate. Total iron must be greater than the dissolved iron. That is ferrous or ferric strands, right? Very good. This is the validation. Actually, data, you just validate the data, right? Next. I have called this thing total solid is more than total dissolved solid. Total solid, total settleables or suspended solids. COD is always better than BOD. Total coliform always bigger than fecal coliform. Total iron is equal to ferrous plus ferric. Phosphate, total phosphate is always greater than phosphate. EC is always greater than TDS. Total oxidizing nitrogen, that is there. Total oxidizing nitrogen is better than nitrate plus nitrite. Hardness is equal to calcium hardness plus magnesium hardness. These are the general checks that I have already discussed. Next. Conditional check, total dissolved solid specific conduction has a correlation. pH and carbonate species, pH and free metal concentration. Dissolved oxygen and nitrate. If pH is less than 8 point, in nitrate is more, dissolved oxygen less, because nitrogen stabilize taking the oxygen. pH is less than 8 point, then carbonate must be 0. It is a bicarbonate, right? Alkalinity is bicarbonate. Next. So again, ionic balance, interrelation of parameters, selection of outliers. Outliers is there. If you have a good database, for a long time database, you can see it is mean plus minus 4 sigma. If it is more than that, it is outlier. You cut it, right? General 10 interpretation, you have to go arithmetic mean, geometric mean, and median. Arithmetic mean and median is sufficient for this thing. And dispersion of data, what is the range, standard deviation, and variance? If standard deviation is very high, then you will know your frequency must be there. Generally, you can go the percentile value 25th, 50th, 70th. If more than 50th percentile value is more than the standard, then you are rejecting. This should not go to that place. It will create a problem, I'm telling you. So train temporal means year to year. Spatial means one station to other station. This is what is the trains. You can go for analysis of variance and non-parametric analysis. Exceeding factor in percentage. That means exceeding factor means your standard in below and over above this is the denominator is a standard and this denominator is up this your observed value. If observed value is more than the standard, then it is a violation, right? Next. One correct estimate is equivalent to 100 expert opinion. You know, my German friends always telling me, Basu, why you are going to meeting when you have no data? Because they are telling there must be a data, there must be an observation. Then you can go for expert opinion. Intuition is good, but intuition is not a science. You have to experiment it, right? Clinical trial is good that this fellow is possibly have a diabetics. Then you test it. Then you confirm it and then give the medicines. Clinical trial is a first things which has been observed for a long time and making it. Then you have a little bit intuition. Yeah, these are the things are happening. This looks anemic. So say that what is the hemoglobin? That is this question. This is the way you have to take. Thank you very much. Thank you.